no cheese, Gromit. Not a bit in the house.
Poor Wallace does not have any cheese. And you will discover that to make this cheese, fermentation is going to be used. Therefore, in this chapter, we're going to talk about fermentation. Number 7, fermentation. The first thing we're going to look at are the different applications of fermentation. We'll see different examples, and for each process, we'll have to state where fermentation occurs and what fermentation is used for. The first example we're going to look at is how bread is made. You've got different steps. To start off with, you need water, yeast, and cereals. There's a mixing process, resting process, kneading process, and rising process. I'm going to circle this one out. I'll explain why afterwards. Then there's this shaping. Then there's a second rising process. Again, I'm going to circle it out. Baking, cooling, and eating. Why is this rising process been circled out? The rising steps have been circled out because in these ones the mixture is going to rise, therefore gas has been produced. If gas has been produced, that means there has been fermentation. Therefore, we can circle out step 4, step 6. This is where fermentation occurs. And what we can also say is that the production of CO2 will imply that the mixture rises. So the reason fermentation is used is to make the mixture rise. The second example is beer. To make beer, you need to use cereals. One kind of cereals, for example, barley. These grain cereals are going to go through different steps: mashing, filtration, boiling. Aha! Fermentation. That's what we're interested in. So step B is fermentation. Yeasts are used to do fermentation. They're going to use up the sugar, which is within the cereals, and CO2 and ethanol will be produced. So the CO2 will give this bubble effect to beer, and the ethanol will have, well, two purposes. The first one is drunkenness. Some people will be happy after drinking drinks with alcohol. And the second purpose is preservation. The fact that ethanol is present in the drink, that will kill off certain microorganisms. Our third example is yogurt. Yogurts are made from milk. The transformation of the milk will be done by two types of bacteria. Some will have a long shape and the others will be in a circle shape. And you've got these lines of circled shaped bacteria. These bacteria are going to do fermentation and they're going to produce acid. This acid is going to have two main functions. The first one is it's going to coagulate certain proteins and fats, so it's going to make the yogurt solid. The second purpose is that the acid molecules is going to lower the pH, make it more acid, and some microorganisms can't survive in acid solutions. Therefore, it will kill off some of the microorganisms. So if we sum up the two purposes, the first one is solidification, the milk becomes solid, and the second one is preservation. This diagram will help us illustrate how the fact that the yogurt, by being more acid, will be preserved longer because there are less microorganisms. This very simple graph shows that the growth rate of different microorganisms will depend on the pH of the solution. Some microorganisms are said to be acidophile, and they will live in pHs which range from 1 to 5. Others are said to be neutrophile. They will prefer pHs from 6 to 8.5. And then you have alkalifile microorganisms, which will prefer pHs from 7.5 to 11.5. Therefore, when the pH decreases, so we're going towards this part of the graph, the neutrophile microorganisms and the alkalophile microorganisms will be killed off. That's how yogurts are preserved longer, because of the acid molecules which are produced. Our next example is cheese. To make cheese, you also need milk, and you need specific bacteria. So you have the bacterial culture here. You have the milk there. And first step, there's going to be fermentation and the production of lactic acid. So where I've shown the arrow from bacterial culture using milk to lactic acid, this is where fermentation takes place. Just like for milk, there are two purposes of fermentation in this case. The first one being solidification, and the second one being preservation. So same thing, the acid molecules will change the organization of the proteins, the fat molecules, and make it solid. And same thing, the fact that the pH is lowered, that will kill off certain microorganisms. Our next example is wine. Wine is made from grapes, and there are different steps: crushing, pressing, clarification. And there are two steps that we're interested in, is fermentation with skin, fermentation without skin. Depending on the wine, the process can be slightly different. So there are these fermentation steps, and just like for beer, there are two main purposes. The first one being drunkenness. Again, for some people it will make them happy, so they drink for that. I forgot to say it earlier, but obviously you must drink in small quantities because there can be counter effects. And the second one is preservation, the fact that there is ethanol that will kill off certain microorganisms. Now then, we've detailed the different examples, we said where fermentation takes place and what was the purpose of using fermentation. We are now going to have a look at 7.2, two different types of fermentation. These are the two different types. We're just going to state the key things. One can, for example, be done by yeast. The other one can, for example, occur in human muscles. But there are other examples of organs and organisms which will do certain types of fermentation. In both cases, we have glycolysis, which will transform glucose into 2-pyruvic acid or pyruvates. Same thing here. In the process, ATP is produced, and so is NADH. The difference is, in the next steps, there are different enzymes which are going to do different things. In this case, so you remember enzymes speed up reactions. In this case, we've got pyruvate decarboxylase, and in this one, lactate dehydrogenase and alcohol dehydrogenase. These different set of enzymes which will be present in these either the cytosol of yeast or the cytosol of mussels will imply different reactions. In the first case, the pyruvic acid will be transformed into acetaldehyde, and this acetaldehyde will then be transformed in ethanol. So this first one is alcoholic fermentation, where ethanol is produced. And the second one, pyruvic acid, will be transformed into lactic acid. In this case, lactic acid is produced. If we link with the examples we've just had a look at, beer and wine will be using this first type of fermentation, alcoholic fermentation, whereas cheese and yogurt, they are made using lactic acid fermentation. We are now going to have a look at the overall equations you need to know for the baccalaureate. You can register all of these, you need to know these equations. The first one is lactic fermentation. In lactic fermentation, pyruvate NADH2 will be transformed into lactic acid and NAD. Lactic acid, we have it here. The second one is alcoholic fermentation. In this case, pyruvate NADH2 will be transformed into ethanol, CO2, and NAD. So this one here is ethanol. You need to know these equations by heart. They can be part of knowledge-based back questions. The two other equations you need to know for the baccalaureate are the following. The first one is the overall equation of respiration. This is what it looks like. We've got a similar case as for photosynthesis, where this equation is slightly more complex than the one you learnt in S4. In S4, you learnt that glucose plus 6 oxygen gave 6 CO2 plus 6 H2O. So you had the simplified version where you take out the 6 water molecules on each side. But scientists, just like for photosynthesis, by using specific isotopes, they were able to say that this is the complete equation. So this is what it looks like. You need to know it by heart. And the second equation is the overall equation of glycolysis. We're just going to state which molecules there are. In the reactants or substrates, we have glucose and NAD. And in the products, we have 2 pyruvates and NADH2. This is what you need to know in terms of equations for this part on respiration and fermentation for the baccalaureate. To end this video, I've found a quite nice song based on Irish music, which explains a little bit about beer and fermentation. I hope you enjoy it. Bye for now. Goodbye.
In the year of our Lord 1811, on March the 17th day,
the feast of St. Patrick,
I will raise up a beer, and I'll raise up a cheer for Saccharomyces cerevisiae. Yes, here's to brewer's yeast, that humblest of all beasts, producing carbon gas, producing acetaldehyde. But my friends, that isn't all, it makes ethyl alcohol.
yes, that is what the yeast excretes, and that's what we imbibe. Oh, anaerobic methylation, alcoholic fermentation,